All right, awesome. Well, thanks so much. I will try and be mindful of time. This is a little bit off the path of my usual soapbox, so we'll see how the timing works out. But yeah, my plan for today was sort of introduce the concept of what a foundation model is and what transfer learning is and how we use them in general in AI and machine learning, and then talk about applications in astrobiology specifically and some of the special considerations we need to think about when we're applying some of these techniques to astrobiology and how that differs from how we might typically do that in machine learning writ large. So a little bit of background on me. So I'm at the University of Georgia. I'm in the Department of Marine Science, and I do AI for environmental microbiology sort of in general, but my background's very much in marine microbes and understanding microbes that live in weird environments. I sort of cut my teeth on marine microbiology and hydrothermal vent ecosystems. Done a lot of collecting seawater from the sides of various ships out in the middle of nowhere. Microbes that live, deep under the seafloor. So microbes that live in really extreme environments that are these wild microbes that have very poorly understood functional capabilities and that have a lot of novelty that we don't understand very well. And one of the sort of unifying themes, I'd say, of that work is that microbes in different environments have really different functional capabilities. And we see this over and over again anytime we measure it. And everywhere we go, in every environment we look in, we discover something new. And that makes it really, really hard to predict it because the data that we have to try and predict these weird microbes doesn't really reflect the functional capabilities of those microbes. So I've spent a lot of my career kind of figuring out how do we apply AI to these systems in ways that are generalizable and that capture the information we really want to be able to predict. So microbes, and I should caveat this whole talk that I'm going to have a pretty strong biological bias in all of my examples, but there's, I think, a lot of these concepts translate to, whether you're looking at geochemical information or molecular information or other things like that. So microbes, we know, scale up to ecosystem and global scale processes here on Earth. Over evolutionary time, that really shapes the geochemical conditions on a planet. Anywhere that life is going to live, that's going to be true, and that's certainly been true on Earth. From the sort of hadal, lava-filled methanogenic Earth that we think of from 4 billion years ago to our, nice oxygenated habitable Earth today. And of course, these days we're thinking a lot about how we can harness biology to enable space exploration and also to potentially identify how we would identify an inhabited world elsewhere, right? So, going into all these weird environments in the ocean has a lot of analogs to extraterrestrial exploration and astrobiology. It's both really hard and expensive to access the ocean. It might cost $40,000 a day to charter a boat to go and take your samples. And so because of that, our data sets tend to be really small and really limited. And we're trying to capture these really complex systems of biology interacting with environment and these global scale processes. So you're trying to model something very complex with very small data because it is so hard to access that. So, I'm not sure how expensive it is to operate the ISS every day, but I'm guessing it's more than 40,000 a day. And to, go explore Mars is going to be even more than that. So I'm thinking about how to get the most out of the precious data that we can collect for our extraterrestrial data and also our ocean data is really important when we think about applying AI to these systems. So we have these special challenges for AI and astrobiology and environmental microbiology in general. And that's most of our biological information, most of the information we want to be able to model is in these environmental systems in rare environments that are hard to access, and we get really small data sets. And so it becomes really hard to generalize models to novel environments. And so you need some special approaches to think about how am I going to generalize my model to somewhere totally new that I haven't seen before. And that's at an extreme when we're thinking about astrobiology and going to an entirely different world where all the examples we have are from here on Earth. And also trying to capture really complex systems with very little data. And so I mentioned this, but I want to kind of hammer this home, is that the training data that we use for any AI model when it comes to biology is not representative of the thing that we're actually trying to model. And this is a huge problem for all of the models that we train. It's very, very hard to generalize to new environments. And that's because, you go into the environment, we can sequence what's there, right? So we can get the DNA, but there's not very much that we know about who they are and what they're doing. So less than 1% of microbes can be cultured. In this figure on the right, I'm showing microbes at the level of the phylum. So that's one step down from kingdom. It's a really broad category. And anything in black here is a phylum that's uncultured. So you can see there's entire phyla of microbes where we have no cultured isolates, right? So we're missing a huge chunk of biological diversity in that we can, interrogate in the lab. So the other way that we do this is we take our sequences that we can sequence in the environment and we try and say something about what function it can do computationally. So conventionally with bioinformatics, you take a sequence and you compare it to some sort of reference database. And this reference database is, all the proteins that we've ever experimentally annotated ever. And what you get when you do that is a pretty similar picture. So anything in this red bar, this is a global survey of ocean samples from the Tara project. Anything in red is unknown function. And so you can see, especially in the deeper environments, meso here is the 600 meters deep water, you have the vast majority of your genes are entirely unable to be annotated. And I will say that if you compare this to reference proteins that have experimental evidence of their function, this number goes down from like the 30% to 1 or 2%. So we have a really big bias in our reference databases. And as I mentioned, those reference databases are very biased. It's not a random sample of 30% of genes that can be annotated, right? It's a lot of E. coli genes. And there's huge misannotations in our reference databases because you end up getting error propagation through these databases. So as many as 40% to 80% of your annotations in these blue bars can be wrong. So we're working with really, really limited window on what we know about the world of biology, which means our training data that we're trying to plug into these AI models to do, say, functional prediction or whatever it is you're trying to do, is really unrepresentative of the thing we're trying to model. So we need to come up with ways of generalizing to new environments, new functions, and thinking about how to do that, often at a performance cost, but I think, worth it in terms of enabling biological discovery. So this is a real bottleneck in AI and in environmental microbiology in general, in all of biology, I think, but especially in astrobiology, right, where you're thinking about even weirder environments, even more novel environments. And so there's a lot of potential in foundation model training to be a key enabler for generalizing to new environments, making predictions about entirely novel things, and I'll talk about why that is. But they need to be generalizable. And the emphasis is really on that generalization. And we need a lot of sort of special approaches to think about that. Okay, so all of that sort of caveats aside, let's talk about what a foundation model is, right? So what is a foundation model? So there's very simply, a foundation model is a big model that's been trained on a lot of data that learns information about that sort of data modality that underlies sort of all of the world of data, right? So it's learning sort of fundamental features of that data. So that data might be text, it might be images, it might be videos, it might be biological sequences, it might be a mixture of these different modalities, but your foundation model is trained in a way that it learns sort of these fundamental features of the biology, and then you can adapt that foundation model that's been trained on an enormous amount of data to a bunch of downstream tasks that you might actually care about being able to do. And because you've learned all of these useful features upstream of that, you can use much smaller data sets to then do these tasks that you actually really want to do. So, in these examples, this is a pretty seminal paper on like what a foundation model is. And they focus mainly on, language and text and images and, the things that we've seen such huge progress in foundation models in recent years. So a foundation model, it's trained in what we call self-supervised way. So basically you make the label out of the data itself, which means you have basically infinite training data, right? So you can scrape all the text on the internet in the case of a large language model. So something like a GPT model does something like this. And what you do is you mask out a word and you say, the cat sat blank the mat, right? And so you understanding language and context probably have a few good guesses about what word might fit in there, right? And so what you do with the model is you train it to do exactly that. So the cat sat blank, fill in the blank. And in order to do that task, the model has to learn, associations between words, and needs to understand rules of grammar. It needs to learn, sort of the rules of language in order to make a good guess about that. And so you do that on lots and lots and lots and lots of data on this really very simple task. Given this text prompt and a masked word, predicts what that next word should be. But that really enables lots and lots of downstream tasks. So you can do in-context learning of question and answering, instruction following, summarization, all the things that your chatbot is really good at doing. Okay, so this is another way of kind of looking at that. So again, thinking about language models, which are quite analogous to biological sequences, there's text sequences versus biological sequences. You have some large corpus of unlabeled text. You use that to train your foundation model. You do that masking task and try and fill in the blanks. And you train this big foundation language model that predicts this task, that blank, the text in the blank, sees how wrong it is, and over time, over many iterations of that, gets better and better, right? And so, we're not here to talk about transformers. Most of the language models are using some kind of transformer-based model. Under the hood, it looks something like this, where you have many, many layers of these. We're looking at sort of a GPT-style model here, a decoder-only transformer, where you basically take your input words or tokens, they get embedded into the model. They go through these mini layers that learn, what other words to pay attention to in the sentence in order to predict that next word. And then it's just trained on this one objective of predict the next word. And you do this over and over again, and it ends up enabling lots and lots of downstream tasks. And it's very powerful in learning those rules of grammar that underlie language. So, the reason why this kind of works, why foundation models give us this big data performance on small data, is because in order to learn a complex system, like a biological system, you need a complex model. You need a big model. You need something with lots and lots of parameters in order to have enough parameter space to sort of capture that complexity. But in order to do that, you need a lot of data, right? So, conceptually, as you sort of increase your data size, if you're using a small model, there's a certain limit to how well that model is going to do, because it's just not a big enough model. It's not a complex enough model. You don't have enough parameters to capture the complexity of your data. As you increase your model size to, a smaller and then a bigger neural network, you can then really capture the complexity of that system. But of course, if you're down here in the small data world, you'll notice it kind of doesn't matter what model you're using. You just don't have that much data. You don't have enough information to train the parameters of a really complex model. So you really need to get up to this big data space to get the benefit of a big deep learning model. And of course, in biology, we almost always have this small data, right? We're almost always working here. You've gone out into the ocean and you've collected, 20 samples painstakingly over a week that you're trying to glean some information out of. And so what foundation models do is you train this big model over here on lots and lots of data. And and it gives you the performance of a deep model on your small data setting. And the way you do that is with transfer learning. And so we'll talk a little bit about what transfer learning is. So transfer learning is the basic idea that you can take these sort of fundamental features of your data modality, whatever that is, natural language, biological sequences, that you've learned on a huge amount of data, and transfer them, port them over to a new setting. So you take your model with its pre-trained weights, and then you just sort of fine tune it. You tweak it on your labeled data set, that painstakingly collected data set, to predict the thing you actually care about. So that it looks something like this. So, in your traditional setting, if you were just training your model from scratch, right? Like you have your data set, maybe it's a couple hundred data points, and you try and train a model on it, and it probably doesn't do that well because you're trying to predict something that's kind of hard to predict with very little data on too small of a model, right? But the idea of transfer learning is like, okay, I'm gonna take a huge amount of data. I'm going to train it on a different task, this foundation model task, some self-supervised learning task. And from that, I'm going to learn domain knowledge of my underlying data modality, the language of life, the rules of grammar that underlie all of human language. And then I'm going to take that domain knowledge, that pre-trained model that has these weights that have sort of captured this domain knowledge, and I'm going to just take that as a starting point. And then I'm going to fine tune my model weights based off of my little data set and get much, much better performance than I otherwise would have. And so in the context of language models, it's the same idea, right? So you might have a large corpus of unlabeled text. It's just stuff you've scraped off the internet, and you've done this mass language model task on it to train your foundation model. Then you port this foundation model over, and you have your small labeled data set, say of maybe, biological domain textbooks. And you want to train a language model that really understands, the language we use around biology. And you fine tune that model on your small labeled data set of, this is the biology topic, and you get a lot better performance on the task of either predicting biological language or generating biological language, whatever you're trying to do downstream. And so in the language model world, I mean, it's moved on from this in some ways quite a bit, but it follows this playbook pretty, at least the first couple of steps are the same idea. So you do pre-training on this massive amount of unlabeled data. You end up with your base foundation model. Then you do your supervised fine tuning. So supervised, I think you guys had a supervised, unsupervised seminar already. So if you don't know what that is, go back and watch that one. But you have basically your small data set with your labels associated with it. And you fine tune your model to then predict the actual thing you care about. In the case of language models these days, and I'll mention this again a little later on, they also do quite a lot later on to align those models even more to the preferences of the user or the downstream task of the user. And we'll touch on that again. But if you ignored this, you can get a lot of benefit with just these first couple of steps: your self-supervised pre-training and your supervised fine-tuning. And the reason why this is because deep learning models are capturing these complex functions, these complex sort of rules underlying their data modality, but they do it in sort of layers of complexity, right? So, if you take an example of a foundation model that's trained on images, there are certain things that are common to all of images: edges, corners, stripes, geometric shapes. You don't necessarily need to have a really well curated labeled data set to understand what a curve is. You can take images from anywhere to learn what a curve is. And so you learn that in these sort of earlier layers. And then tends to be in the later layers where you learn these really task-specific features, things that are, really specific to, I'm trying to train my model to recognize dog faces. And that might be kind of later on in the model where you capture those really specific features to your prediction task. And so you can take these earlier features, these fundamental features of edges and corners and stripes and curves, and apply them to an entirely new labeled data set of, say, here's my cells. I want to predict whether there's a tumor in this cell. And even though you've trained on nothing that really looks like a cell, pumpkins, elephants, dogs, cats, you can, with a pretty small data set that some grad student has gone in the lab and collected all these microscopy images, make a prediction about whether there's a tumor in those cells by simply porting over, like, I understand what a curve is. I don't have to relearn that. I can use my precious experimental data to just learn the things that I care about to predict whether there's a tumor or not in this cell. So in biology and astrobiology, I would argue there's a lot of applications we might want to do transfer learning for, right? So if you can imagine some great biological foundation model where we've trained it maybe on this language model task, something very similar. So a DNA sequence is pretty similar to a sequence of text, right? It's a sequence of nucleotides. You can mask that out, and from that you'd have to learn how codons are organized, how genes are organized, amino acid preferences, things that are really relevant to things like function and evolution and, things that you care about for downstream biological prediction tasks that we actually want to understand. So from this, you have this foundation model that learns a contextual representation. You might then fine tune that to do, or you're training that to do hidden nucleotide prediction, so that masked nucleotide task. And then you can imagine transferring that knowledge for all sorts of things. And these are just a few examples. So at the nucleotide prediction level, that's things like fitness prediction. You might want to generate novel de novo sequences based off of some condition set. Or you might want to take the embedding, here's a whole sequence, and here's my sort of latent representation of that sequence. And I'm going to predict things like function prediction, like what function does this gene do? Or whether this gene is going to be expressed given some condition set, something like that. And I'm sure, whatever your work is, there's plenty of applications that you could think of as well. But there's this sort of dual place where you might want to do that, either at the contextual representation level, at this sort of latent representation of a sequence that captures all these sort of fundamental features of biology, or at the single nucleotide prediction stage. Okay, so that's kind of what a foundation model is. That's the idea of transfer learning. It all sounds great, right? It works really well for language and images, and it works okay for biology right now. But there are a lot of sort of special considerations when we think about biology, especially in environmental contexts, and especially in astrobiology contexts. So I introduced this a little bit earlier, right? We have these special challenges. We need to generalize our model to extremely novel settings, and we need to model really complex things with very little data. And so we need to think about, what do we need to do from a modeling perspective, from an AI perspective, to be able to do that, right? And so I'll talk about each of these kind of in turn. First, I want to talk about model generalization and how we can generalize to novel environments. So there are starting to be a lot of biological foundation models out there that are, trained to do very different things. The sort of most famous ones are probably the protein language models, so sort of the BERT class, the AlphaProt class, ProGPT2, that sort of thing. Also ones trained on DNA, so genetic ones, genome level ones. But there's a lot of these out there. The point here is, we're just looking from 2019 onwards, right? And you can see, there are a decent number, even starting in 2019 and even in the past year, we're just seeing an explosion of these. So there's, a lot of interest in AI and biology in general, but most of these models are not optimized for environmental settings and will not generalize to them. And I will tell you why. So just to take a couple of examples, really famous examples of protein language models that have then been trained to do 3D structure prediction of proteins. So AlphaFold is a really famous one, right? Just got a Nobel Prize. ESM2, there's actually an ESM3 now, is another famous one. ESM2 takes just protein sequences and is then transformed, is then fine-tuned with a bunch of tricks to turn that into a 3D structure. AlphaFold takes the protein sequence, but also a bunch of other information like a multiple sequence alignment, other information around the protein and sort of our domain knowledge based off of bioinformatics to then do 3D structure prediction. So these require a lot of domain knowledge, right? They require full-length protein sequence. If you've done anything in an environment, you know that when you go into the environment and you sequence the environment, you get these short reads, right? They're like 100, 150 base pairs. And in order to get a full-length protein, you have to assemble it. And I will tell you, if you've ever tried to assemble a metagenome, the amount of your reads that are going to assemble into a full-length protein, depending on how diverse your environment and how deeply you've sequenced it, is going to be like between 5 and 25% of your reads will assemble into a full-length protein, right? So you're going to have a hard time doing that in environmental sequences. And it means that our protein reference databases are very biased towards, things we can have in culture, things that we can assemble because they're very abundant, things like that. In some cases, it requires additional protein information. So in the case of AlphaFold, that's true. And my personal hobby horse is, we do a lot of trying to predict structure from sequence for some reason. And structure is a proxy for function. If what we care about is predicting function, I don't know why we're not just predicting function. But I won't go down that rabbit hole right now. But the point is that these models are really requiring training data sets that have a lot of domain knowledge, that are these full-length protein sequences that are not representative of the world of biodiversity, right? So if we want to make a biological foundation model that's generalizable to environmental sequences, there's a few things we can do. So we're going to use a case study from my lab to sort of illustrate this. So first of all, we know that reference databases are extremely biased in terms of what proteins are there. It's the things that have been heavily studied historically. It's the things that we can sort of easily get into culture, things like that. But we have lots and lots and lots and lots of environmental sequencing data. We have exabytes of metagenomic DNA at this point, over 1 million metagenomes. And a tiny, tiny fraction of those are represented in those protein databases, maybe 1% of them. So we can train a biological foundation model on this unbiased environmental sequencing data, which does represent the world of biodiversity pretty well here on Earth. You can find novel things if you go into a truly novel environment, but most sequences, I would say we're coming pretty close to seeing most of the biodiversity on Earth. And so if we're going to train a biological foundation model, we need to really think about how to represent this biodiversity in that model. And so that's what this looking glass model does. So basically, instead of taking your protein reference database and using that as your training data set, what if we just take all of the environmental microbes we've ever been able to find, and then evenly sample our training set from across this prokaryotic tree of life? Break that down into sort of read length sequences that look like environmental metagenomes, and then train our biological foundation model on that data set using that same task, right? That same mass language model task. And it turns out that does a pretty good job of capturing fundamental features of biology, and it also makes training a lot faster. You can use a lot less data to get the same information out of your data. So what I'm showing here is training time from left to right, and then the training data set size. The blue colors are randomly sampled sequences, so stuff that isn't this uniformly sampled DNA, but is just randomly sampled sequences. So that's going to be biased to like really overrepresented microbes, things that are really abundant, lots of delta proteobacteria and E. coli, right? Things that we have lots and lots of genomes for. And you can see, as you get more data, as you go from light blue to dark blue, you do better. You get better training performance. But, it's not doing all that great. You can get as much as 10 million sequences, and it's not doing as well as this pink line, right? This pink line is the uniformly sampled data. So this is, we took a genome from every class at the class level in the prokaryotic tree of life. This is the GTTB taxonomy, and use DNA from each one of those genomes. And so you can train a lot faster because you're using a smaller training set and capture more information than you otherwise would. But you also need to make sure that when you're doing training, you're evaluating your model with data that looks different enough from your training set. So the way that I like to talk about this is that you're evaluating on an out of distribution test set. And that's really important if what you want to do is ultimately deploy your model in the environment where it's going to perform well in an environment where you know the biology of that environment is not going to look that much like the training set that you trained on. And so the other sort of trick on the data side that you can use if you're trying to get to capture these really generalizable features of biology is to split your data set so you have your pool of data that you've uniformly sampled, right? And then you're going to split it into your training, validation, and test set, right? So your training set is the one you're actually training your model on. You have a validation set that you're seeing sort of how am I doing in terms of generalization. You can do your parameter optimization on that. Then you have your test set that you don't look at how your model's doing until you're completely done training the model. And when you do that splitting, you split them based off of sequence identity or taxonomy. So what we did is we said, okay, we've uniformly sampled these genomes from across the tree of life at the class level. We're going to make sure that no genome from the same class is in the validation or test set than is in the training set. So nothing with a lot of high sequence identity is shared between your training, validation, and test set, right? So your average nucleotide identity between training, validation, and test is really low. And so that's going to ensure that whatever rules you learn in the training set, as long as you're not overfitting to your training set, and you can watch that as you see your training go along, you will know that when you deploy that into a new environment where you're potentially encountering completely new microbes, completely novel biology, it's going to do a pretty decent job of understanding the fundamental features that are common to what it's on the training set and what it's now trying to understand in a new environment, without getting too far down the road of just trying to memorize what we have in our reference databases, right? Because they're very unrepresentative. So it's going to be a lot more generalizable. So that's basically what Looking Glass did, right? So it took our read length DNA from across the periodic tree of life on that training set that shares no microbial classes with the validation or test set, did this self-supervised learning task of a mass language model to predict, what nucleotide should be here, given the context of the sequence around it. And through that process, that model learns to embed those fundamental features of biology, those functionally and evolutionarily and environmentally relevant features that are common to biological diversity across the tree of life. And so when you capture all of these features, you can then fine tune that model to do all sorts of interesting things. So it can do functional prediction pretty well. It can detect distant homologs that have really low sequence similarity, things that wouldn't be caught with our conventional bioinformatics methods, differentiate different environments, sort of identify important nucleotides for particular functions. You can predict optimal temperature of an entire protein from a read length fragment of DNA, which I think is pretty cool. Reading frame, pathways of evolution, novel oxidative reductases. So this is a particular functional class we were interested in. But the point being that you can fine tune this basic biological foundation model to do a lot of pretty disparate prediction tasks, right? And you, of course, are evaluating that on a completely separate validation set and training sets for your fine tuning that are quite different from your training set that you use for the foundation model training. And it turns out when you do this that the features that you capture when you do biological foundation model training, accounting for this biodiversity, you end up capturing a lot more generalized and useful functional features of your sequences that then generalize way better to your new settings, to new functions. So this is we're just looking at the performance in the test set. So this is a slightly different training set. I won't go into how we formed it, but it's a similar principle where your test set looks very sequence dissimilar from your training set for functional prediction. And you'll see Looking Glass, the latest iteration of it, is capturing this accuracy is kind of a measure of sort of the functional features that are inherent to the model. It's not doing full functional prediction, but you can see it's a lot higher than a lot bigger models, biological language models that have been trained on with a lot more powerful computers and a lot more data, I will say, by a decent margin. But you still have a problem where if you're looking at like a rare functional class, in this case, we're looking at glycosyl hydrolases, which is a functional category we particularly care about in my lab. But you see this for any sort of rare functional class. You do have a big performance drop off, and Looking Glass is still capturing better features than the other models. But you still have a problem where your class imbalance, where really rare data is pretty hard to capture because you just don't have anything that looks like it in your training data. So the message here is that you can get really far with model generalization by using some of these tricks around data splitting and data curation, but it doesn't eliminate your training data bias. At the end of the day, we have really limited data, and we would benefit a lot from having better data in a lot of cases in biology. But I do want to accentuate that we can use these models to really enable novel discovery of novel biology. So what we're looking at here is one of these functional prediction models from an older version of Looking Glass that was trained to predict oxidoreductases, whether a read length sequence I get from the environment is an oxidoreductase or not. And so you can see our classifier is this one on the left. It predicts for 20% of the sequences in an environmental metagenome from these various samples from the ocean. Our oxidoreductases, about 80% of them aren't. When you compare that to these two sort of conventional bioinformatics functional annotation tools, you can see there's these big gray bars, and these are the ones that don't annotate to anything in reference databases, right? So we have these samples that are really dominated by what we call microbial dark matter, things that don't look like anything we've seen before. And that's very, very typical of environmental samples. But in the case of an AI model, it doesn't matter if you've seen it before, right? Like you're taking a sequence and you're making a prediction. So that's the real sort of potential of AI applications in biology, is that it's reference free. You're not dependent on these known knowns of things we've seen before, in theory, if you can make really generalizable models, right? And so if you apply this oxidoreductase model to these samples, you see, trends in depth and latitude in the percent of oxidoreductases in a sample as you apply them, right? And this is very typical in the ocean. You will see a latitudinal and depth gradient basically no matter what you do, right? But the point is, with this oxidoreductase classifier, you see a certain trend, right? If you compare that to your conventional bioinformatics tools, those trends either go away or even reverse using your conventional annotation tools. And so there's a lot of information we're losing in these conventional approaches where we're throwing out 30% or throwing out 70% of our data to begin with, and the 30% we have annotated we think is misannotated by like as much as half of it. That might be pretty garbage data that we're then trying to interpret downstream, and it really limits us to this world of known knowns, right? And so the power of AI applications in biology and in astrobiology is really in potentially transcending that dependence on reference databases, on our world of known knowns, and potentially discovering things that are very different from what we thought they were when we look at it from a different approach, right? Okay, so that's in terms of model generalization. There's more we could talk about, but I can't be here all day, right? So the other problem we have is we have a lot of complex systems we're trying to model, and we have very little data to model it with. And so even with a really good generalizable biological foundation model, when you do your fine tuning, you need to think about how to do that in the best way to sort of get the most performance out of your very limited labeled data set. And so I want to talk a little bit about a few approaches that you might take to do that. Of course, it's not an exhaustive list. But one way that we kind of talk about this often is few-shot learning. So with a small data set, what we really want to do is we might only have a few examples. And we have our biological foundation model or our foundation model, and we want to fine-tune it to predict the thing that we care about with very, very little data, maybe just a few examples. And so in few-shot learning, what you're usually trying to do, so this is an example where you're trying to classify like a new species of penguin, right? What you're usually trying to do is learn this similarity function. So I want to learn a similarity function that maps a novel query, like a new image that maybe is an entirely new label or that we haven't seen before in some other way, closer in feature space in my model embedding space to the most similar training examples that that model saw during training. Or you can think about mapping queries that are farther from dissimilar samples. You can imagine doing this in a different way. There's a lot of ways that you can approach learning this similarity function, but that's the basic idea, where if your new novel looking thing is going to look most similar upon inference or upon training on your few shot examples to things that it should already be mapping towards, you're going to be able to get a much higher performing model than you would if you just sort of fine-tuned it brute force from the beginning. And so there's a lot of approaches to learning that similarity function. I'm not going to talk about all of them because we're getting near the end of the hour, but you can imagine a few different ways you might want to go about that. There's also this concept of parameter efficient fine-tuning. So big foundation models in general, they have a lot of parameters, right? Not all those parameters are going to be the most relevant to the thing that you're trying to ultimately predict when you're doing fine-tuning, right? And so this is another one of those taxonomy trees of various papers that have been published for parameter efficient fine-tuning. There's a lot of ways you might want to do this. What they're all trying to do is only fine-tune on a subset of parameters, a smaller number of parameters, to get the same performance than you would get if you trained like that whole big foundation model with all of its maybe billions of parameters. And so there's a few ways you might do this. You might train something. This is sort of the adapter-based fine-tuning approaches where you might train something kind of separately and then combine it with the foundation model. You might select a smaller number of features within that model to fine-tune the ones that are most relevant to your thing that you're trying to predict. Or you might want to do something sort of in between that where you're merging you're trading a smaller number of parameters on the side and then merging that with your foundation model in order to produce your output. So there's, a whole lot of approaches that are in, various classes that you could go down that rabbit hole if you're interested in it. But all of them are kind of unified by is there fewer parameters that I can train to more efficiently get my predictive model that I care about on my small amount of data. The fewer parameters you need to train on a small amount of data, the sort of more statistical power each of those data inputs is going to have, right? And the last one I'll mention in terms of getting good model performance with smaller data is kind of specific to large language models at the moment, but I think a lot of people are trying to think about how could you apply something like this to other domains. And so a lot of these large language models these days have moved from these sort of masked language models, here's my input, here's my prediction, to these reasoning models. And what that basically is you have a large language model that's been pre-trained, and instead of just spitting out the answer directly, it does a reasoning process before it gives you an answer. So if you go to ChatGPT and you ask it a question now, if you're using one of their reasoning models, it'll take a little longer to give you an output. And that's because it's doing this reasoning under the hood. And there's a lot of there's a whole sort of a class of reasoning approaches that are, ever growing, of course. But you kind of think in a chain of thought way, some sort of thought process, right? Which is similar to how a lot of us think, is I think is the intuition. In order to get to your final answer, you kind of go through the steps to build up to your answer. So if you're trying to predict something really complex, that can be really helpful, right? And what we find is that much smaller models, if you give it more thinking time at inference time, can outperform much larger models that are, going straight to the answer. And so, the way to think about this, if you wanted to think about how to apply it to your domain, is, how do you go from a non-reasoning model, like a regular large language model, to a reasoning LLM? Is you have to come up with a certain amount of these traces, right? Like you have to have some kind of training data set of, here's my prompt, here's my thought process, and here's my output in the end, right? But the number of examples that you need to do this is pretty small. You can have, maybe hundreds of examples of your PNS without the thought process, and maybe tens of examples of the thought process, and get to and if you have a pretty powerful LLM to begin with, you can learn a subdomain in a pretty short amount of time, a pretty small number of examples. And they're doing this usually with reinforcement-based learning to learn how to do reasoning. So, this is one that's a little bit less intuitive, I think, for something like biology, but I think there's a lot of sort of lessons to be learned there about how to use less data to get more output out of an existing foundation model. Okay, so to sort of wrap things up, I think AI is a real key enabler for astrobiology because it really breaks our reliance on this world of known knowns of, we have one example of life here on Earth. So if we're going to try and go to another planet and say, here's the environmental data that I can measure, what's the likelihood that something lives on it? You really need to think about how to generalize and extrapolate beyond the distribution that you've always seen. And there's, a lot of progress, I think a lot more progress to be made on doing this with AI. I think it has the biggest potential for enabling us to do this. And we've made some decent progress on generalizing to novel and rare environments, by using some of the approaches I've just discussed today. But I do want to just close on one last sort of hobby horse of our lab at the moment. For something like functional prediction or a lot of the examples I gave today, a lot of the time we're going to need more data at the end of the day. So I gave that example of the sort of general functional prediction versus the rare class functional prediction and the big performance drop that we have. And part of that is that we just have massive data gaps for biology in general and definitely for astrobiology. And so I think, there's things that I think AI can inform us on to fill in some of those data gaps in a really smart and, sort of most efficient in terms of time and expense way. And so that's another thing we're working on that's a little bit off topic, but I think is very relevant to making generalizable foundation models, is creating those benchmark data sets that we need with careful experimental design that's AI guided to enable those downstream prediction models. All right, and with that, I want to thank all the Hoarfrost Lab members that are fantastic and have contributed to a lot of the sort of lab-specific work that I talked about today, and take any questions.
Awesome. Thank you very much, Adrienne. This is fantastic. This is also perfect. I just want to say with helping, as you're trying to do, with helping to bridge that gap between the LLM techniques and the astrobiology applications. So I think everything you listed here will be very beneficial to the community going forward to be able to come back and try to look at these different techniques and see what is relevant. So yeah, let's get into any questions. If you have a question, you can either feel free to raise your hand in Teams or just post in the chat and we'll go from there. We'll just give people a second. Caleb, go for it.
Hi, thank you. That's fabulous, fabulous talk. Really, really informative and interesting. And actually, I wanted to try to ask a question you sort of answered part of my question with your last slide, where you talked about figuring out where the most information gain is to be made in essentially an unknown environment. And this may be a very difficult question to respond to, but I was also thinking about, let's say you drop down on Mars, or you get into the ocean in an icy moon. The environmental data that you may be able to acquire, even if it's sequence data, may be degraded, it may be noisy. Can this process of looking for the most information gain, the measurements that would give you the most information gain, also be used to try to figure out, how to best handle degradation of the fundamental things you're trying to measure in the environment? So let's say a short sequence is somehow degraded. There's noise. Is that something you've thought about?
Yeah, I mean, so I think there's ways of like trying to clean up data where you could, it's difficult if you're thinking about sequence data on another planet, because like if you're seeing sequence data on another planet, I don't know, you got a lot to think about there. But yeah, I don't know if the relevant thing would maybe be like some sort of environmental measurement, or like some sort of spectrometry measurement or something, where you might have degraded data. I think there's a lot you can do in terms of data cleanup, where it's like automated data cleanup. You can predict what part of this data is noise and what part is not. And the trick would be, in order to train like a noise reduction model using your Earth-based data, is you have a similar problem where you're going to be in a very different environment with potentially different patterns of degradation once you go to Enceladus or whatever it is. And so you'd have to think again about can I hold out a data set that's going to look most like the environment I want to deploy it in, and have that be the test set. And that'll give me kind of the best sense of how well that's going to do. You can also do things like have a human in the loop, and try and like automate 80% of it, 80% of decision making. And that might work on a place like Mars, where you're only waiting maybe 15 minutes to have a human look at it, as opposed to, that being basically impossible on Jupiter. But yeah, I think that would be my general approach, not knowing a ton about the particular data that you're talking about, being more unfamiliar with that. But yeah, that's my instinct.
No, thank you very much. And I had only a vague thought about data, so but that's very, very helpful answer. Thank you.
Howie, you had your hand up next. Why don't you go for it?
Yeah. Can you hear me okay?
Yes.
Okay, good. So my question is, I've been paying a lot of attention to the mass spec data from the plumes on Enceladus lately, and hopefully we'll see similar data from Europa when Clipper arrives a few years from now. And so would the AI be useful in this setting, do you think?
Yeah, I think so.
Does that work at all?
Vaguely. So I mean, I know there's mass spec data coming out of it, right? And what are the things that you guys are trying to predict out of that, would be kind of my question.
Well, would be a clue to the existence of life there. And the mass spec data produces these fragments, and I think it would be, well, NIST has made a bunch of fragment libraries, and I think fragment libraries of the data from Enceladus would be useful.
Yeah. Okay. Yeah. So, I mean, I'm not a mass spec expert at all, but yeah, if you're getting mass spec, like spectra that are telling you something about like the molecular composition of the plumes, right?
Yes, exactly. Yes, more so
yeah.
than it's like very, it feels to me very connected to the actual physical reality of what you're looking at, rather than spectral data, for example.
Right. Yeah. And so you're trying to say, given this sort of molecular composition, or probably more accurately, like some sort of spectra, that's your actual raw data, what's the likelihood of life being able to survive here or already living here? And yeah, I mean, I have a lot of thoughts on like how specifically to do this. I've been trying to get someone to fund me to do this for a while. But I think, I mean, I think the thing is you have to map between environmental conditions and biology as we know it. And then use some tricks to sort of generate what biology should exist under a certain environmental condition set. And then you have to evaluate whether that's real biology or not, right? And the trick is in figuring out how you differentiate between real biology and not real biology. But yeah, that's the short answer, since I know we're going over time, but I could talk about that all day, actually.
Oh, sure. Well, yes, that's a very interesting topic. And I had one other quick question. So it seems to me that on Earth, we have environments that are parallel to the icy ocean worlds, and that our icy oceans are not nearly well enough studied, and that further study of what's going on in our icy oceans would really help this astrobiological quest.
Yeah. Yeah, I mean, I think a huge data limitation that I have specifically for like habitable metrics is finding environmental conditions associated with environmental metagenomes that look like the data we're going to collect elsewhere. And so more of these analog systems where you have data that is specifically the data you're going to collect from a rover or your instrument up there associated with the metagenome would be a huge data gap to fill, in my opinion.